Welcome to Beck, the Behavior, Evolution, and Culture Speaker Series. It's wonderful to see you all here. Katrina has brought a good crowd of people today, and it's really great to see some new faces as well in the room. Welcome. If you're new to the speaker series, we meet every other week during the academic year. We have a website, bec.ucla.edu. If you're interested in this talk, there may be many other talks coming up that you'll be interested in as well, and next year. So check out our website, and if you want to, you can subscribe to the Beck listserv through the Get Involved tab on the website. So let me just give a preview of what's coming up for the rest of the quarter. We have Annie Wirtz from UCSB, who will be visiting us in 2 weeks on May 12th. Her talk is called How Infants Learn to Eat Plants and Why They Might Not Want to. And then our final talk of spring quarter will be Professor Erin Riley from San Diego State University. Her talk is Roadside Primates, Balancing Risks and Rewards in Anthropogenic Environments. So those are both going to be interesting talks. Please join us for those as well. Today, it's my pleasure to introduce Professor Katrine Whiteson from UC Irvine. We met recently at a symposium at Cal State Northridge for John Patton, who some of you might have known, who is an important member of the evolutionary science community in Southern California, who passed recently. Katrina collaborated with John and his students, and that's going to be part of her talk today. She does research on the microbiome, and I also want to mention, because I'm not sure if you're going to talk about it, but in our meeting beforehand, we were also talking about research that Katrina does on using phages to control infections from bacteria, basically, that are antibiotic resistant. So another whole area of really interesting research that you might want to ask her about afterwards at the coffee or in meetings later on. So her talk today is May the Fiber Be With You, Gut Microbiomes from a Remote Amazon Village to the Undergraduate Lab. Please join me in welcoming Katrina.
Thank you.
Oh, and cards. Does everyone have a card before?
Oh, that's right, yeah.
Let's pass these down. These are part of the activities.
I'll explain it. So yeah, it's really nice to be here. Thank you so much. Can everybody hear me okay? Great. Yeah, you guys have a beautiful campus. I'm really lucky to be here. And I just want to start big picture and think about the kinds of problems that focusing on our microbiomes could help us solve. So, it's more than just studying what we have in our microbiomes, which are the communities of bacteria, viruses, fungi, and other microbes too small for the eye to see that live in any environment, and our body's a really important one. And if we can optimize our health through the microbiome, we could, for example, really improve our cancer treatment outcomes. That's a really exciting area of research right now, where we know that people who eat more fiber have sometimes double the progress-free survival. That example comes from immunotherapy patients who had melanoma. We could really reduce our dependence on pharmaceuticals that are intending to improve our cardiovascular health. So some of the big pharmaceutical blockbusters like statins and even Ozempic, arguably fiber triggers the same pathways as Ozempic. So we could use fiber to have some of the same outcomes that have been so exciting with Ozempic in the last couple of years. And in general, if you're eating fiber, you're protecting the community of bugs that live in your colon. So if you take antibiotics, you're giving those bugs a place to hide out. And so I'm going to walk you through all of this in the course of the talk, but these are the kind of big picture goals that we're after. So I'm joining you from UC Irvine. We're right between Los Angeles and San Diego, and we're near the ocean and the mountains, just like you guys are. I know you can relate to that. And I have a couple save the dates here. We're having our SoCal Microbiome Symposium that rotates around Southern California is going to be at Caltech this fall. And we're also having a symposium about the microbial dark matter at the ranch in Laguna. The company Zymo is sponsoring that, and I'm helping organize it. So that's going to be really fun. And before I get too far, I really want to acknowledge my lab. And actually, Catherine McCarthy was a student in our lab before she became a grad student here at UCLA. She's even depicted here. We all went out to Catalina Island one day to visit the USC Wrigley Reserve, which I can really recommend that. They'll even, the boat ride is on them if you organize something out there. So that's a really fun thing to do. And I just really want to acknowledge the team of people who have made all this research possible, especially Andrew Oliver led a lot of the work I'm going to talk about. In the last few years, I've put a lot of energy into teaching in our teaching kitchen at UC Irvine. So we have a really beautiful kitchen. It's like Great British Bake Show style. And I have monthly webinars, and I'm actually teaching a freshman seminar right now. And I just have a picture from last week that is so good, I have to share it. So, and my family made a lot of fun of me for taking this picture, like, why wasn't I helping, you know? But Chef Jess, my partner who I've been teaching with for all these years, had it under control. I could tell, you can see that pot lid is about to land. The fire went out in a second. So I knew I had a moment to like take a really great picture of her saving the day. And a chemistry student, Donovan there, with an electric stove managed to start a fire, a 3-foot flame. All that's in that pot is a little olive oil and some onions, and somehow he managed to get that combusting. And yeah, Chef Jess is like, nobody believes me that you can start a fire with an electric stove. Well, there you go. You do not need an open flame. And it all worked out just fine. And a lot of the students in this class had never chopped an onion in their lives. They're 18 years old. And so yeah, it's kind of interesting. You don't really need to like go to a UC to learn how to chop an onion necessarily, but but actually it's been really fun, and you know, I think we all enjoyed that moment. Okay, so just to back up, I should define microbiome before we get too far. And microbiomes are the communities of bacteria and viruses and fungi and other microbes that live in a particular environment. And I've been working on this in this area for about 15 years. This field didn't exist when I was a grad student, so I switched into it after grad school, actually. And when I first got into it, it was right when DNA sequencing became a lot cheaper. And so these projects became more accessible. Although, to be honest, the microbial ecologists studying the soil and the oceans have been using methods for a century to study these communities. But now we have a lot of data describing the microbial communities in human environments. And a lot of it is correlation data. So we now have correlations showing that there are certain types of microbes associated with obesity and cancer and autoimmune diseases and brain chemistry and some types of neurodegeneration. Even vaccine efficacy is very much tied to our microbial community composition and all the immune physiology that's associated with that. And so it's a really fun time to be a microbiome scientist. And now we have this big challenge of taking these correlations and making sense of them and figuring out how to use interventions and not just observations to make use of the data. So most people have very unique microbiomes. So most of the genetic differences between you and anyone else in this room are encoded in your microbes. We have crazy diversity in our microbial communities. Microbes can live in every environment. No oxygen, no problem. I mean, keep in mind they were on the planet well before we had oxygen, and they are responsible for the oxygen on our planet. And so anyway, microbes are very creative and have very impressive diversity. That's why so much of the unique diversity associated with each person comes from the microbes. You share a lot of microbes with your household. Sometimes when people ask me, how can I get a healthy microbiome? I'm like, well, get a healthy roommate. Maybe that'll be on the Craigslist ads in the future, I don't know. And some of the most dense microbial communities on the planet are in human microbial communities. So I have a plot here showing density of microbes in different environments. And you know, some parts of our digestive tract have pretty low microbial density in our stomachs. It's not that there's no microbes, but it's pretty acidic, and there's only a few kinds of microbes that can really take that. But as you go up, you'll see that colon and dental plaque samples are some of the densest, even more than soil, which is famous for teeming with microbes. And then just keep in mind, anytime you have bacteria, you probably have 10 times as many viruses infecting those bacteria. So in our guts, we have high numbers of bacteria, and then usually several different types of viruses that are able to infect those bacteria. So there's no such thing as a cell that doesn't have viruses infecting it, and that's been true throughout all of evolutionary history, and the same is true for the microbes in your body. And so this is the era when I was a postdoc first getting into this field, that this idea at the time felt really crazy, that disease could arise from a disturbed microbial community. And it's so interesting, it's not something we're cultivating. We're so careful to vaccinate our babies, but we're not like actually making sure that every baby gets a healthy microbial community at the start of their lives. We're just starting to figure that out. But probiotics are an $80 billion and very unregulated industry. So that's not something that we're doing very intentionally as a society. And that's something that I could really see changing. And I've had a lab at UCI for 10 years, and it's always been this like long term, I imagine us being able to cultivate that. And that's still the sentence, that's not happening yet, but we do have ideas for which bacteria babies need, for example. And so, and ask me more about that. I have a couple slides about it. So here's the backdrop. This is probably the plot that convinced the National Institutes of Health to fund the Human Microbiome Project around the year 2007. And it shows how we had this really impressive decrease in infectious disease in the second half of the 20th century. And this is a big part of why human lifespans expanded so much in the 20th century. And by the way, a lot of other health things have gotten better since the 1950s too. I don't mean to be gloom and doom. I mean, everyone was smoking, and heart disease and lots of other health issues were much worse in 1950 than they are today. So there's been huge progress on many fronts. But there also has been this kind of mysterious rise in allergies and autoimmune diseases. And this plot was made back in 2000. Now I would add some kinds of cancer, Parkinson's disease, maybe Alzheimer's disease, autism. There's several other conditions that we now understand are connected to our microbial exposures. And so an important hypothesis behind these ideas is that our exposure to microbes has been changing because of all these great public health measures, in part sanitation, vaccination, the use of antibiotics. And so we really are changing how our immune systems develop. And so I know this is a really broad audience, and I can highly recommend this book, Missing Microbes, where Marty Blaser, who is one of the fathers of the hygiene hypothesis, talks about how an average child in the United States is getting 18 courses of antibiotics by the time they turn 18. And so we're doing this experiment on humanity kind of unintentionally, where we're really changing our microbial exposures, and we're not really fully understanding the consequences of that. Okay, so now for our activity. So I have these recipe cards, and I think there's still a few if you need one. And so I'm gonna ask a few questions, and this is something I do in my big micro classes at UCI. I was very inspired by my co-instructor, Travis Wiles, and a student in my lab, Ritwick, made these cards. And so I'm gonna ask you a few questions. So, yes or no, have you ever taken antibiotics? Okay, actually there's a couple no's in this audience. That's very interesting. It's pretty rare to get a no. I got my daughter to her 13th birthday before she had to take antibiotics, which of course I'll take. I mean, I believe in antibiotics if you need them, but it's pretty common to get antibiotics in our society. So mostly yeses there. Okay, how about, have you taken antibiotics this year? Okay, so most people are saying no. That's kind of interesting. And this is obviously voluntary, no pressure to answer these questions. That's interesting. And then, okay, do you know anybody with allergies? Mostly yeses, that's interesting. And it would be very interesting if we could go back 50 years in this same room and invite all your colleagues then and ask them the same question, because they might have had a different answer, right? Like, I think my kids can't bring peanut butter to school because we have to be careful about peanut allergies, and that was not an issue when I was a kid. And I think that actually that's getting better, so that might be better in the next few years. Okay, and then here's another question. Do you know anyone with diabetes? And I mean either type 1 or type 2, type 1 being autoimmune and type 2 being more lifestyle, or everyone. Yeah, that makes sense in our society. So those are some of the conditions that we're talking about that have become more common in the last half century. Now, if you look around the world, you can see pretty important differences between the types of microbes that are present in the gut. So this is data where the x-axis of this plot is age, and the y-axis is all the different types of bacteria using a sequencing approach. And so you'll see in red and in green are samples from Africa and South America, and in blue are samples from the United States. And so you'll see at the beginning of life, everyone is starting out with a pretty small number of microbes, which is normal, and then it starts to flatten out around ages 3 to 5 as people are eating solid foods. But then you'll notice that in the United States, everyone is plateauing off at much lower numbers, more like 1,200 with this way of counting the different kinds of microbes, whereas the people in Africa and South America have many more different kinds of microbes. Another way of looking at all of the different microbes from people around the world comes from this really nice meta-analysis from a group at Stanford, the Sonnenberg group. And here I've just summarized, there's samples from about 35 countries here, but nobody can read 35 colors, so I've just summarized it here a little bit. And so on the left side of the plot are samples from less industrialized parts of the world, and on the right side of the plot are samples from more industrialized parts of the world. And each point on this plot represents one fecal sample. And then there's a really nice trick on this plot where they show you the abundance of different bacteria in that exact sample right underneath. So let's take a look at the less industrialized part of the world. So not so many Bacteroideaceae, but really high numbers of Prevotella. And Prevotella, by the way, are bugs that are famous for carrying the genes that break down plant fibers. And they're typically associated with what we would consider a healthy gut, although healthy is almost a philosophical word there, so I'm not sure how to define that. But I'm just pointing out that you'll see some really big differences. Look at the abundance of all these bugs in the less industrialized parts of the world. And now let's come over here to the US and Europe and the more industrialized parts of the world, and you see this huge abundance of Verrucomicrobia. And these guys are mucus-degrading bugs. So my interpretation of this plot is that people eating processed food are having all the food get absorbed early in their digestion, so our colon is devoid of the substrates that microbes like to eat. And instead, those microbes are eating the mucus that lines our gut. That mucus is what is between you and sepsis. So like, you don't really want that mucus layer to be getting broken down. However, these bugs, they're also called Akkermansia, are sold as a probiotic with Halle Berry and Tom Hanks, making it look really good. And they do have positive health associations. So microbiology is complicated. It's, calling something good or bad is a very hard thing to know how to do. Maybe this is a great optimization of what microbe you want in an industrialized gut. That's a science question, but I'm just showing you this data, which I think is really interesting, that in the parts of the world where people eat more plant fiber, their guts are full of plant-degrading bugs. And the parts of the world where we eat Big Macs and granola bars that we think are healthy but are just actually a bunch of sugar and processed wheat and stuff, then we're getting all these bugs that break down mucus. Another interesting trend is when immigrants come to the United States, you can see how their microbiomes shift. So you just got familiar with Prevotella in light pink in this plot. And this is a study of immigrants coming from Thailand and living in Minnesota for different numbers of years. And you can see that the people in Thailand have a majority Prevotella in their guts. And then with every 5 years that people spend living in the United States, they lose more and more of that Prevotella, and it becomes replaced by a bug that's common in the United States, Bacteroidetes. And so after 30 years, the immigrants from Thailand resemble US-born people. So this is a trend that's happening across the world. And so we're losing the microbes of human history. And it's not so random what we're colonized by. You can actually follow human migration patterns by looking at the mutations that are collected in human-associated microbes. In some cases, human migration patterns are better reflected in the bacterial mutations than they are in the human genome mutations. So it's really like our microbes are a big part of human history. We have evolved with them. Every creature on Earth has been evolving with microbes because the microbes got here first, right? So this is a really big change in everything about our biology. And it's interesting because it's all happening, we can't see it happening. And so there's been some really interesting efforts to try to preserve the microbes of humanity. And the Global Microbiome Conservancy is part of it. And actually Ben in our audience works with the guy who took the photograph here, and he can tell you the story. And there's a documentary I really recommend called The Invisible Extinction, where they describe efforts to make a microbiome vault where there's groups going and working with different local communities to preserve the microbes in each place so that as we go through more industrialization, we don't lose those microbes forever. And just to focus on infant microbiomes a little bit before I jump into our own lab's data, if you look at the early life microbiome development in industrialized and less industrialized places, you see that through human history, most babies have been dominated by specific species of bifidobacteria that are really good at breaking down breast milk. And about 15% of the energy in breast milk is basically fiber. They're called human milk oligosaccharides. And we are losing those microbes too. So an average baby born in the United States will be basically having none of that turquoise bifido that can break down the human milk oligosaccharides. And so that's really interesting. Even if you're breastfed but you don't have this bifido, it really changes how your immune system is developing. And this is one probiotic that, in my opinion, has really strong evidence behind it. And this was all developed at UC Davis, and they have a company, Evolve, and a probiotic, Avivo, which was just isolated from a healthy Davis baby. So do we want to recolonize humanity with one Davis baby's bifidobacteria? That's kind of a philosophical question too. But this bifido can break down those human milk oligosaccharides and protect the baby gut from being colonized by bacteria that might have difficult immune consequences. So there's big studies going on, like there's a study of 1,000 babies that are at risk of developing type 1 diabetes in 5 different places in Europe right now, and they're giving all the babies this bifido, and so we've got to wait like 10 or 15 years to find out what happens, but this is an area that I think has a lot of potential that I'm really excited about. So, so what can we do about it? That's a great example. Probiotics are something we can use to shape the microbiome. So are prebiotics, which are basically fibers and other molecules that promote healthy microbiomes. We can also use fecal transplants, and there's now 2 FDA-approved products that are getting fecal transplant to have a more official role in medicine in the United States. They're both in the treatment of Clostridia difficile diarrhea, which is caused by taking antibiotics usually. And then another one that is not the focus of my talk today, but I'm happy to talk about it, is phage therapy. And that's the idea of using bacterial viruses as alternatives to antibiotics. So like the enemy of my enemy is my friend. So I'm going to talk about our fiber projects a little bit. And it's really interesting to figure out how we can fill our fiber gap in the industrialized world. Most of us are getting about half of the fiber that's recommended, which is the recommendation is 25 to 35 grams per day, which is half of what people are eating in a lot of parts of the world without even trying, where their diets are more plant-based. But an average person in the US is getting something like 10 to 15 grams of fiber per day. And the fiber comes from places you might not expect. So I've been doing fiber interventions in my undergrad classes, and I'm going to tell you about that. And I think a lot of people initially think they're going to have to eat a lot of salad to get their fiber intake up, but you'd have to eat like a lot of salad, like 25 cups, to get that much fiber. Whereas some foods are very dense in fiber. So you want to be looking at the carbohydrate to fiber ratio, and it's really high in avocados, beans, berries are surprisingly high in fiber. Chia seeds, lentils are in the bean and lentil category. These are foods that you could quickly fill your fiber gap with. And the idea is that, looking across the gut, most of the bacteria in our microbiomes are anaerobes that live in the colon, the last part of our digestive tract. And if you eat processed foods where the fiber has been removed, then there won't even be any nutrients making it to the colon. Whereas if you eat higher fiber foods that still have the fiber intact, then you're going to have some of your starches and fibers make it all the way through the digestive tract down to the colon where the anaerobes live. And why do you want them there? Well, those fibers get fermented into molecules that are the favorite molecules to fuel the cells lining the epithelia layer of your colon. So you have this one layer of epithelial cells lining your colon, and those cells love butyrate and other short-chain fatty acids. Whether you know it or not, everyone in this room, if you're a human, has smelled butyrate. It is one of the major gaseous products of digestion. And yeah, this molecule is what the epithelial cells want. If you feed those cells glucose, their mitochondria get all unhappy, you know, your risk of getting cancer goes up. Those cells, their metabolism does not work as well if they don't get butyrate. So if we starve our colon of those cells, that's the reason that, that it's a problem. So this is one of my favorite quotes from John Steinbeck. And I don't know if there's anybody in this room who's a member of the Rancho Gordo Bean Club, but if you know, you know. This is the newsletter quote this is like a quote that's at the top of the newsletter every quarter when you get your package of beans. And this was written with the idea that you could use beans to protect yourself from starvation in winter. And so in the novel Tortilla Flat, Steinbeck describes how the abuelitas would go out to the fields and ask the farmers, like, hey, do you mind if we pick over this field that's already been harvested? Maybe there's some extra beans out there. And then they could grab those beans with the goal of getting 400 into the multi-generational household to kind of get everyone through the winter. So we're in a really different time right now, but both for climate and for health reasons, if we could lean into plant-based proteins and fibers like beans, it could really have positive impact on our health, on our immune systems, and also on our climate. So let's just have a look at black beans real quick here. So, the lunch had chickpeas. The ratios would be very similar for chickpeas. So if you take a serving of black beans, half a cup here, there's about 20 grams of carbs, and nearly half of them are fiber. So like if you were dosing insulin for this half a cup of black beans, you could subtract the fiber from the carbs. Those fiber will not hit your bloodstream and get your blood sugar up after you've been eating. They will stick around and persist into your colon and get fermented into all those molecules that are the favorite fuels for the cells lining the colon. And they're also a good deal. So this is a recent Trader Joe's image. And frozen berries, for example, just as nutritious, if not more nutritious as fresh berries. They get frozen right on the field at the peak of their freshness. And they are very high in fiber. And they're sweet. It's so ironic, but that's a really good source of food. So, you know, I don't want to be making a message that's inaccessible, but dried beans and frozen berries are some of the more accessible things I can think of. You do need to have like electricity to cook dried beans, it's true, but I mean, I've even had people participate in studies who don't have a kitchen. You just have a crock pot, you can plug it into the wall, you can make yourself beans that way. It takes a little bit of planning, and to be honest, I think most of our nutrition messages in our era are not really science or education based. Like, we know we should eat more plants. That's not the issue. A lot of nutrition programs just like try to say it more slowly that like you should eat more plants. But everybody got that message in second grade. Like, why are we spending $1 million for Netflix to make a documentary to tell people stuff they already heard 4 million times? Like, people are not having a hard time understanding the message that plants are good for you. Everybody got that one decades ago. That's not why people are struggling to eat enough plants. It has to be appealing. It has to be doable. I don't even know if cost is really the reason it's inaccessible. That is a reason that's often given. I think it has more to do with just making it work in your life. So if you want to eat more fiber, You just have to think about what you actually do every day and what you actually like and pick one thing. It could just be, I'm going to eat chia pudding at lunchtime every day. And then on the weekend, you buy some berries and you stew them. So then on weekday mornings, you can just like make yourself chia pudding. It'll take you 1.5 minutes per day. And then at that moment when you're starving and you would otherwise go out and buy a donut, you would eat something that has fiber in it. Like that's the kind of change I think would make it work for people. Okay, so we got some more questions coming here. So do you eat beans regularly? Yeah, most people don't eat beans around here. That's true. I'm seeing that. What about chia? Chia's had kind of like a social media moment. And the texture's not for everyone. So that's another thing. You got to pick something you actually like. Okay, what do you prefer, chia or beans and lentils? All right. Maybe it's more beans. That's interesting. Cool. Another top question I get is, can I use supplements? Does it have to come from food? Like, what if I just eat Metamucil every day? Will that solve my fiber problem? So one more question. Do you take fiber supplements? So a lot of people take fiber supplements. And so, actually I'm not against using supplements. Figure out something that will work for you. It's better to get it from fresh food. Okay, now here's another one that might seem subtle. What do you prefer, Metamucil or Miralax? Like, I know a lot of families where they're giving their kids Miralax every single day. Okay, I'm not getting a lot of Miralax in this audience, which is good because Metamucil is plant fiber. Metamucil is psyllium husks, so it's coming from plants, whereas Miralax is polyethylene glycol, and that really can be damaging to your microbiome. So if possible, I would try to avoid relying on Miralax on a daily basis. For example, if you compare, like they've done studies where they give mice either Metamucil or Miralax when they're getting antibiotics, and the mice getting Miralax get terrible infections because the Miralax does not protect their microbiome in the same way. So in our classes at UCI, we've been getting students to eat high fiber diets. So now you guys have a lot of background about why we did this. And we've been publishing both education and science related papers about all the results. It's been very fun. People giggle a lot. We work hard to make it all anonymous. So here's the sample drop off, which is like in a different room than where the class is. So you can be discreet and go drop off your sample. And yeah, it's been really fun. Andrew Oliver, I mentioned, was a grad student in our lab, and he had to be like a camp counselor, getting everyone to drink enough water and everything too. And so here's our study design. We collected 3 fecal samples in the first week, and then we had everybody eating high fiber for 2 weeks and collected 3 more samples towards the end. How did we get them to eat so much fiber? Well, in this particular intervention, we used whole meals from the company Thistle. I looked at all the meal prep companies from around Southern California, and the Thistle meals had between 15 to 20 grams of fiber usually, so we got 2 meals a day like that and then just gave people advice beyond it. Here's some of the things they leaned into when we took surveys. So this is the number of people who said yes to eating the different foods listed here. And the first time we did this, I have to say I really learned a lot. I had never tried to look at nutrition labels so much from a perspective of fiber, even though I care about it a lot, but this really forced me to look at it. I didn't know that avocados are high in fiber. I thought of the creaminess of avocados as coming from fat, like you always hear about the healthy fats. But fiber is creamy. Like if you make lentil soup and it's got like a creamy texture or like hummus, those fibers can be actually quite creamy. Like soluble fiber is creamy. Like oatmeal, when you cook it fresh and it gets kind of gloopy, or chia pudding, that's fiber. So anyway, these are some of the foods that people were really leaning into. There was a little bit of fiber supplemental bars, which tends to be inulin that they put in there. But people were eating a lot of mostly whole foods. We really did have to talk about constipation. That's might be the opposite of what you would expect from eating fiber, but fiber without water is concrete, so you have to be sure that you're drinking enough water. And so, big reveal, did they actually eat more fiber? And you'll see in the first week, There's students eating 0 grams of fiber in some cases. And like, if you look at what they were eating, it's like pizza and beer, you know. But on average, they were very close to the US average. In this class, it was closer to 20. I just got new data back from last quarter, and it was 13 grams before and 26 grams after in an intervention with chia seeds. This quarter, we had the whole food that I was talking about, and you can see we got up to 40 and 50 grams of fiber a day in weeks 2 and 3. So that's, in my mind, a really big success. I still see the students sometimes, some of them are around, and I think it really is a message that stuck with them lifelong, so that's pretty cool. One of the big signals you can see from these microbiomes that we sequenced from the fecal samples is that each person is quite distinct. And so each column in this display of the data shows you the microbiome profiles from a different person. And the gray ones are before the intervention and the red ones are after. And so, you know, big picture, the biggest signal comes from which person donated the sample. And that is true across every microbiome study. A lot of them are cross-sectional, so then you just have 90% variance unexplained. Here we are lucky we have longitudinal samples, so we can see a little bit about what happened. And you'll see there's some people where just even from the back of the room, you can see this turquoise bifido expanding a lot after the fiber intervention. But it's somewhat individual. And so that was just an important message that they're all very, each person is quite different from each other. Going into it, I actually really thought that we were going to see an increase in the diversity of the microbiome from eating more plants. I showed you the data from around the world and why we were thinking that. But remember, it's just a 2-week intervention. We were very inspired by other dietary interventions with just 2 weeks where they had seen health relevant like decreases in polyp-like formations in just 2 weeks of eating a high fiber diet. So if I'm honest, I really did think that the diversity was going to go up. But in reality, we didn't see much of a change in the overall microbial diversity. If anything, we might have seen a little bit of a die-off. And now that I've had years to reflect on this data, it makes a lot of sense because you're changing things. And so the old regime is going to have to, you know, move on, and then you need time to build up the new regime. So this is essentially the start of a revolution, and probably we should be doing 1-year studies. But how many 1-year studies are there out there? Like really almost none. It's hard to keep an intervention going for that long. And yeah, so another thing we saw was that the bacteria that is the most changing with the intervention is a bifidobacteria, different than the one that I was talking about with the babies. We actually did a more focused analysis on the bifido, and each person carried about 8 different strains of bifido. And overall, that was the biggest signal of what increased with the high fiber diet. And it makes a lot of sense. Bifido are also famous for carrying genes that are good at breaking down plant fibers. A student from UC Irvine in my ecology colleagues lab, Jen Martini, she led a big meta-analysis of every fiber intervention data set she could find. It was right when the pandemic hit and we had to go home and couldn't do lab work. So she emailed 800 people to get their data, and only about 10% of them responded. And still, it's a wonderful analysis. And you can see that we are not alone in seeing the diversity go down. So this is a measure of bacterial diversity, and you'll see that across every single one of these studies, and each of them also are pretty short weeks intervention, you see a decrease in microbial diversity. And we're also not alone in seeing bifido as the bug that's going up. So 3 of the top 5 bugs here are bifido for responding to fiber. So I'll summarize my fiber intervention studies here just to say that we do see significant shifts in the microbial communities when people eat fiber just for 2 weeks. In terms of life advice, really I would say it'd be smarter to start more slowly and build up to it. Think of your gut like a muscle, and you wouldn't just go to the gym and try to lift really heavy things tomorrow. You would build up to it over a year, and that would be the big message I would share from a health perspective. So out of this audience, I think it's kind of hard to know, to be honest, so it makes complete sense if you don't know. But who in this audience thinks they probably eat 40 grams of fiber per day? All right, there's some yeses. That's cool. Then what about 20 grams of fiber per day? Cool. That makes sense. And that's, I mean, I think a lot of Americans are getting 10 to 15 to 20 grams per day. Here's another thought. Do you eat extra fiber when you're taking antibiotics? Okay, there's people who know about that. So even the New York Times is making articles in this direction now, which is really cool to see. And I really agree with this article where they talk about, you know, if you had to pick something to do when you're taking antibiotics, I think some people might jump to the idea of taking probiotics, but actually taking fiber is going to help you a lot more, because then you're going to keep the existing diversity protected and going, rather than trying to replace it with some random probiotic in a package. Okay, and then just last couple questions. Would you be willing to participate in a project like this and have to donate samples? That's cool. Yeah, honestly, despite all the giggling and some reluctance, most people have really been great participants and have enjoyed it. And then, have you ever tested your own microbiome? Have you ever like sent your sample to a company and, okay. So there's like maybe 3% yeses, I'd say, so not 0. Well, it's actually pretty hard to know what to trust if you do send your sample out to a company for sequencing. And here's another New York Times article that I really agree with, where they talk about how each company has their own process for analyzing the samples and their own way of presenting the data. And so you can take the same sample and send it to 3 companies and get 3 totally different answers. And that's not really what we aim for in science. We aim for reproducibility. And so that's a major challenge in our field. And actually, I'm part of some of these efforts. I'm on the steering committee at NIST, which I hope will continue to exist, where we are talking about microbiome and metabolome standards and how to help our studies have reproducible results. Okay, so I have my story about Kanambo and the Amazon as my grand finale, and I hope I have enough time. I understand if people need to get going. And so, yeah, I'm excited to share with you this really fun collaboration. I had the opportunity to work with John Patton, and my student Andrew Oliver led the analysis that you're about to see. And John Patton was an anthropologist at Cal State Fullerton, and he had a field site in Kanambo, Ecuador since the early 90s. And, you know, people in this room know more about it than I do, I think. But I met John about 10 years ago, and we started brainstorming ideas for projects. And this was a really hard thing to do, to collect samples and preserve the microbial DNA down there. And so he was just a phenomenal scientist. He passed recently, and we had this memorial where I got to meet a lot of his colleagues, and it was really fun. And so this group of people are, you know, responsible for all the samples you're about to see, and it includes several master's students from Cal State Fullerton who were living down there for the summer, and they really are the ones who did a lot of the work that made this possible. So in Kanambo, Ecuador, people get a lot of their nutrition from a beverage called chicha, and it's made, the women in the household will chew root vegetables that they've been growing, yucca, and they'll, there's an example of the mash that they're creating in this chicha boat over there. And they'll chew it up and they'll spit it back into a vat and allow it to ferment for a day or 2. And it gets to have a bit of alcohol in it, and everybody is drinking chicha. Kids, everybody, and they're getting a lot of calories, like 2,500 calories a day from chicha. I'm kind of looking at Clark here because I feel like he knows more about it than I do. This is something I learned about to get to participate in this project. And then there's kitchen gardens. I hear the favorite snack of the kids is to intentionally leave out some veggie scraps so that you get some insect grubs growing. And then when your parents are off doing something else, drinking chicha or something, then you start a little fire and you roast the grubs, and apparently it's just delicious. So that's like the kids' favorite snack. And so, yeah, so I got to, here's an example, or here's a map showing you where Canambo and the Canambo River is. So we had 3 major questions that we were hoping to answer by studying the microbiomes down in Canambo. Just big questions like what microbial taxa are present. But then we collected samples at a household level so we could answer questions about cohabitation and how the microbiomes in different households looked. And then we could also compare those microbiomes to other cohorts like our undergrads at UCI. So we collected samples from 28 different households. We had about 100 fecal samples and chicha samples and slightly fewer saliva samples. And so it was a lot of samples. And I've done a lot of projects, and when you have this many samples from a environment where collecting and labeling everything is a challenge, often my experience of working with doctors is that it turns into a nightmare with the labeling and stuff. But this was a very professional operation, so I just cannot thank the team enough. We preserved all the samples in a buffer from the company Zymo that preserves the nucleic acids but kills all the microbes. So that made transport really easy. They're stable at room temperature. And it also meant all the biosafety issues and crossing the border with the CDC permission, et cetera. All of that was much simpler because of that. Okay, so just big picture, what are some of the microbial taxa that we saw? And now you guys are familiar with this kind of plot because you saw one earlier from the different international cohorts. Now I'm showing you chicha samples in pink, fecal samples in brown, and saliva samples in kind of a turquoise. And so here, if you look at the fecal samples, you'll see that some of the bacteria that are quite abundant are ones that we saw in those other less industrialized parts of the world cohorts. So like Prevotella, for example, that's Collinsella. These are bugs that are very enriched in the carbohydrate breakdown genes and tend to be enriched in non-industrialized parts of the world. So this is, if anything, just a common sense check. And then if you look at the chicha samples, you'll see enrichment in Lactobacillus, Saccharomyces, which is famous for helping us ferment nice things. And then saliva samples here are enriched in Rothia is a famous oral bug. And so actually, in many ways, the types of microbes that were present in the saliva samples are very similar to what we see in the industrialized parts of the world too. So the saliva and chicha samples were more similar, which makes a lot of sense. One of our questions was, how is the saliva, which people are chewing the vegetable roots before spitting them into a vat to ferment the chicha? So we were curious to know how the fermentation was working. And we definitely saw a lot of shared microbes between the saliva and the chicha samples. This measure of distance shows you with smaller values that there's more similarity. So, and then maybe reassuringly, the fecal samples were less similar to the saliva samples and the chicha samples. We also saw that cohabitation influences the samples, especially the fecal samples. And so that makes sense because microbes are shared within households, and that's one of the biggest signals that we've seen in other parts of the world as well. And we especially saw that that there was a nice signal of within household similarity in the fecal samples. Big picture, we were curious just what the composition of the microbes was like in the oral samples. And this is shotgun sequence data. So every piece of DNA in the sample had an opportunity to be sequenced. And most of the reads map to bacteria, but in the saliva samples, about 10% map to viruses. And the most abundant taxa is a Streptococcus salivarius, which is also very abundant in other parts of the world. So that really maps with what we expect from an oral microbiome. And then very interestingly, a lot of the reads did not map very well to the databases that we were using. So every time we do an analysis like this, we map the sequencing reads that we get back to a database. And then in many cases, people don't even report how much of their data mapped to the database. They just write the paper with the reads that mapped and don't even mention the ones that didn't map. It might be that 95% of their data didn't hit the database, but they just write their paper with that 5% or 1% and move on. And so that's really interesting, that there's so much to discover, really. And so, so here you're seeing that there's quite a few unknowns, so we don't even know what it is, so we can't say too much about it. That was even more true in the fecal samples. So here's our fecal samples. 95% of the reads are bacteria. The most abundant taxa is Prevotella copri, which is, I would guess, very low abundance in this room. In my undergrad classes, it'll be 1 to 3%, or as a whole class. So this is really stunning to me. This was really exciting. And then the amount of reads that were considered unknowns that didn't map to this initial database that we were using was just a huge number. So there's a lot of things that I can't say as much about because they didn't map to the database. We just saw great enrichment, though, in what are sometimes referred to as vanished taxa. So these are the types of microbes that are more abundant in the less industrialized parts of the world. So that makes a lot of sense, but we really saw that in these samples. If you compare our microbiomes to other cohorts of data that we had access to, you can see that the Canambo samples cluster together with other indigenous samples, whereas the industrialized samples also cluster separately, and with they were together, the industrialized samples. We looked at the what was the main signal distinguishing these samples from the industrialized samples using a machine learning model, and we found that some of the important indicators of the samples in Canambo included this Collinsella, for example, is a strain that is predictive. And Prevotella, which is being renamed, was also one of the ones that was predictive. So yeah, we have some nice signals of industrialization. And if you compare the Canambo indigenous and industrialized cohorts, and actually I'd love advice or thoughts from this room about how to describe the industrialized communities. In previous versions of this work, we had sometimes referred to them as Western, which I think is incorrect. You don't have to be near Roman columns to have your microbiome, which has been affected by eating industrialized processed food and living with other industrialized conditions. That's not really associated with anything Western, but I'm curious. I saw we still have that label here, so I welcome questions about that or comments. But anyhow, you can see that we had really extremely rich diversity. This is a measure of diversity in our Canambo microbiomes, more similar to other indigenous cohorts and higher than the Western cohorts. One subset of the diversity that we're just super interested in are the genes that are important for breaking down fiber. And that was through the roof in the Canambo samples. So there's really high diversity of the genes that can break down fiber in the Canambo samples. And just to pick out some other favorite studies in this group, like here's our UC Irvine undergraduate population. This is the diversity of genes for breaking down carbohydrates in UC Irvine. So clearly much lower than we saw in Canambo. Part of this signal has to do with how deeply the samples were sequenced. And actually, our Canambo samples were not particularly deeply sequenced. This was not a high-budget project. But for example, here's a US cohort, the Human Microbiome Project, that was very deeply sequenced, very high budget. And so that's maybe one reason they still have higher diversity there. My own thinking about this was really influenced by just this. The Canambo project really influenced my own thinking about this famous result from a big citizen science project. And in this project, they asked people if they ate more than 30 types of plants in a week. And that'd be a good question to ask you guys too, so just think about it. Oh, I love it. And so in this project, they show that people eating more than 30 kinds of plants in a week have higher gut microbial diversity. And so then it's really easy to have your mind just explore that idea, like, oh, you need different types of fibers to support different types of microbes, and so the plant diversity begets the microbial diversity. But then I got a really good question from Noel Mueller about my Canambo project, and he's like, well, the people in Canambo can't get 40 kinds of plants in a week. They can't just like go to Whole Foods and buy every kind of veggie. They're mostly eating the yucca, you know? And so, and they have very high microbial diversity. So I think it could be that this result hinges upon actually just that the people who eat 30 kinds of plants in a week also just eat more fiber. So it's not necessarily required that it's a particular diversity of fiber. In a way, that's a relaxing thought, because it's just, you just got to get some fiber in there. Like, don't worry too much about the details. Yeah, assessing the diversity of these samples it's just a real challenge. I was telling you about the databases. And for a long time, this felt like a major limitation in the study. This is a Metaflan 3 database. You'll notice is on the y-axis right here, and the Canambo diversity looks low, but we know it's high, so like, what are we missing here? So we went and did database independent analyses. So for example, we just looked for the patterns of nucleotides in the data without mapping it against a database, so we could just ask on its own what the diversity looked like. And when we did that, then we see the Canambo diversity is like going through the roof again. So that's, that's relaxing. However, anyway, so that became a big message in our lab that, and I love this idea. That's actually the topic of the symposium we're going to have in Laguna in August, is just to include all the diversity in your results. At least report the parts that don't map to the database, so that you are facing the music about which parts of the data are being reflected. If you look in all different kinds of data sets, you'll find that there's a very high fraction that's not hitting the databases, so it's just such a challenge. About 70% of gut microbiomes and maybe 80% in those cystic fibrosis sputum samples, and these are very well-studied human systems, you know. Anyhow, so here's my K-MER analysis where we went and did a database independent look, and here's the diversity going through the roof when we have a database independent analysis. However, the more modern version, now there's another one called Metaflan 4, and I won't go into the details, but feel free to ask me. And actually, this new database is doing a great job. So we have way fewer unknowns. So that's really good news that there has been real progress in sequencing the microbes of humanity, so that when we go and take samples from different parts of the world, we can learn about what's in them. Okay, now I can see I'm short on time. I'm sorry if I'm going too far, but my last story has to do with the fermentation of the chicha. And so we, from a small subset of this is chicha that was fermented by the anthropology students who were down in Canambo that summer. We got time points. So I have the first 4 days of the fermentation, and I'm showing you the abundance of 3 different microbes: a lactobacillus, a yeast, Saccharomyces, and a streptococcus. Streptococcus has to have oral origin. That's the one that's really abundant in our saliva. So you'll see the initial composition is very rich in streptococcus. And then we see the lactobacillus and the yeast start to increase as the days of the fermentation go on. So, we're definitely seeing a signal of streptococcus in the chicha fermentation. We also then did a household analysis of the different streptococcus strains within Canambo, and you see that there wasn't actually such a difference in the strains found in the chicha and the saliva, so that further supports that the streptococcus is indeed the inoculum. But there was one outlier in these streptococcus strains, and that is from the anthropology students. So we could distinguish the streps coming from the anthropology students from California compared to the strains down in Canambo, which makes sense, but that was kind of interesting. Okay, so my conclusions from the Canambo study are that we found their microbiomes are very high in the vanished taxa, The oral streps are household specific, and that the Canambo microbiomes share a lot of features with other non-industrialized cohorts with lots of novel microbes and lots of fiber breakdown genes. Okay, so with that, I'll thank you, and I would happily take questions. I have a few more bigger picture conclusions about fiber and microbiome here, so thank you.
Thank you. Time for questions. Feel free to call on people if you want.
Okay.
Yeah. So just a clarification. So for all your microbiome population, it's based on bacteria, right? You didn't count for in virus and all that kind of stuff.
That depends on our analyses use shotgun sequencing, so we actually can capture fungi, viruses. No, it's not limited to bacteria. Sometimes when we make a plot, we just limit it to the bacteria, but overall, shotgun sequencing should capture everything. The main reason you wouldn't see something is often more about the sample processing steps. So like we use a bead beating step to crack open the cells, and sometimes they're not as good at cracking open fungi, for example.
I want to ask a follow-up question for that. Do you really think the higher of your alpha diversity means good health? Because for most of my studies, we didn't find good health is always associated with higher diversity. So we are really questioning about that conclusion. Where does that coming from?
Yeah, I do not think that more diversity necessarily associates with health, and I think that's especially true in sites of the body outside the gut. So in the gut, we have seen this association between eating plants, health, and high gut diversity. There's other systems like the oral microbiome where higher diversity is very clearly unhealthy. And that's true for the vaginal microbiome and the skin microbiome where, like in the vaginal microbiome, you have just a couple lactobacillus dominating in many of the healthy communities. And so that's very low diversity. High diversity would be an indicator of not being healthy. But for the gut, overall, I agree with you that we should not necessarily think that more diversity is healthy. Yeah. Yeah.
Yeah, hi. So I've always been curious about chicha as a vector for transmission of microbes between people,
Yeah.
right? So firstly, for the oral microbes and the strep, do you see evidence that the genetic differences between strep strains found in different people are rather small, sort of indicating that there's really transmission between people via the chicha? And then do you also see other maybe gut-associated microbes that seem to show up in chicha and then potentially also move between people due to the chicha drinking?
That's a really good question. I mean, I don't think we have enough time points to watch those kinds of transmissions in this particular data set, so I don't feel like I can answer the question from our own data. But overall, I'm sure that there are microbes being transmitted in the chicha. That makes a lot of sense. And so, I know these households have all different kinds of relationships, and so I'm sure some households are drinking each other's chicha more often than others. And so, yeah, there have been other studies that had more time points. For example, the Fiji comp study led by Ilana Brito, she did a really nice job actually where you could see those kinds of transmission events better. But I mean, overall, I agree with you. There has to be interesting transmission going on.
Static data, do you think it's possible to, if you're assembling a map, can you just sort of compare the number of genetic differences in the strep strains and just sort of try to maybe build a phylogeny within a village, within a house, right?
Yeah, we can do that. I mean, we have done that to some extent with the strain analysis from the different households, and you can see that samples from the same household are more similar to each other. Yeah.
Thanks for the talk, really interesting. I was curious, this is a general question, but you mentioned this notion that we all know to eat our vegetables, eat our fruits, and then get our fiber. From a public health messaging perspective, there's been a rise of all meat diets or fiber is not useful for us. And how do you do you have any thoughts on like how we combat that as a society?
Yeah, I've had that question like from so many angles, also from the angle of like Eskimos and indigenous people that have an entirely fish or meat-based diet. And actually, I've even thought about it from the perspective of dog studies where they add fiber to dog diets. And that really got me thinking because you think of dogs as omnivores or carnivores. So one way to answer that question is to look at the physiology of the organism that you're studying. And if they have a long gut and a chamber for fermentation, that suggests that on average in that organism's life history, they have been needing to be able to ferment plant fibers. So that would be my main thought there. In Eskimo microbiome studies, there have actually been these really interesting studies, for example, comparing people in Toronto with Eskimos up in the north of Canada. And believe it or not, they ate about the same amount of fiber. Like an urban Toronto resident eats about 15 grams of fiber a day, and so does an Eskimo from seaweed and berries and things like that. And maybe a little bit more food coming from grocery stores and stuff like that. So, but overall, I think Eskimos are living great. Like you can live a healthy life without fiber. I'm not saying that's impossible, but I think through our human history, we have usually been fermenting fiber in our colons, and some of our physiology depends on that. Yeah.
I really enjoyed your talk. It's so informative, so it's just fantastic. I was wondering, you mentioned the issue of categories that we use to describe different people who are participating in this research.
Yeah.
I was thinking, ideally, one axis would be a kind of dietary metric of what they're actually eating as a kind of way of, because it speaks directly to the core of the research. So
Yeah.
I was wondering if you have any suggestions or have delved into this. I mean, in a Western context where we have nutritional guidelines printed on packaged foods, it's one thing, but when you're working in a remote village where some of the foods might not have ever even been nutritionally analyzed,
Yeah.
that it gets really difficult actually to figure out what people are eating and how much and how it varies. So I wondered if you've taken up that issue and if you have any recommendations. And then if I could just add one other thing to it is that in particular fiber has a role in satiation and your feeling of fullness, and so it can have an impact on your total caloric consumption. And is that something also that you're exploring or not with your research into fiber?
Yeah, really, thank you. That's helpful, good comment. I mean, with regard to collecting dietary information from anyone, anywhere, that's really, really hard. That's part of why my more recent fiber interventions, I've tried to keep it really simple, where it's just like, yes or no, did you eat one serving of like whatever food was in the intervention? And then you do a general dietary survey at the beginning, and you don't really try to get information every day because it's a really big burden and it tends to be pretty hard to get good information. Right now I have a collaborator at IBM and through our project where they're using a bot that calls you at night and asks you what you ate. And then it's like easier, and I just tried it last week and it was pretty easy. I don't know how that would work in a Kanamba-like setting. That is clearly a real challenge. I mean, I think you just kind of have to pay attention to how people are eating and get a general idea. Yeah, that's a good question. And yes, with regard to the satiation coming from fiber, I think that's fascinating. That was what made me make the comment about parallels with Ozempic, because some of these fermentation products from fiber actually do trigger pathways related to the GLP-1 inhibitors. So that's where the feeling of satiation is coming from. But also, like you said, I mean, when you're eating fiber, it's persisting in your digestion and it's making you feel full for longer. So I think it's just a trick we should all know. Just if you're hungry, like make sure you're eating foods with fiber in them. That's going to fill you up. But yeah, good question.
Hi, thank you. I have a question about the dark matters part of that's unexplored. So firstly, you already answered one of the questions, sort of, that I had as you moved through your talk, and I was thinking about, and you showed the two graphs of the, was down the lower left-hand portion, and there was all the sequence reads that were unmapped. And it looks like there were about maybe 15 perhaps different types of strains within that range, right? Because when you compare, I guess your Kamer analysis.
Yeah, I misspoke when I think I jumped the gun when I saw this slide. So I'm just gonna,
Okay.
I'm just gonna. Is this what you were talking about? Because I think I'm.
This was the next part, yeah. Because the
Yeah.
original one was just the mapped reads that were not
Yeah.
mapped, or
So-
unmapped reads to mapped reads.
So it's low richness, high unmapped reads. So this is where I misspoke. I think I thought I already had my Kamer plot here. Anyway, high unmapped reads. And then here's the call to looking at the dark matter. And then here's the Kamers,
Right.
where, the Kamer richness is quite high, even though this one database is stating that the diversity is low. Is that?
Yeah, exactly, right.
Yeah.
So then, so that was interesting, right? But, so then your question got me thinking too, about the genetic diversity of those unmapped reads.
Yeah.
And sort of as an approach to sort of thinking about that dark matter and the relationship between the unmapped reads, you could look at them, right, together from a evolutionary phylogenetic perspective, I
Yes.
think, right?
Yeah, you don't
Um,
need a database to look at them.
Right,
That's
right.
totally true.
And is that something that people are doing and thinking about?
Yeah, I mean, the answer is always yes when it comes to microbiome analysis, because actually there's a lot of creativity, and on average labs work independently, so it's great, but in terms of like a lot of options for how you do it. Like one thought is you can assemble the data, so you take all those reads that tend to be a couple hundred base pairs or less long, and then you make them into, non-biological chimeras in a way,
Yeah.
but like you try to map them all together like a
That was the
puzzle.
first
That
thought that I had.
that's really cool.
Yeah.
Yeah.
Because they're going to be mapped reads that actually are from the same organism, potentially.
Yes, exactly.
100 base pairs long.
Yeah,
Right, yeah.
yeah, and so you can do that, and then that, sometimes you'll end up with big, long, we call them contigs, that are still unknown. But usually, once you start making them into these bigger assembled pieces, they tend to have something map in the database at that point. You know what I mean? Because then you're helping.
Something, yeah.
Exactly, yeah. But I mean, this problem really is getting better, is my overall feeling. But it's still common that people will use a read-based approach and not even mention what didn't map. Like in our mouse, we're doing a mouse Alzheimer's study right now, and the last year's data, only 26% of it mapped, and I think this year 30-something% mapped. We kind of keep track of that each year. And it is getting better, but still, that's like a lot of unknowns, right? So we should probably be doing something like you're talking about in terms of if we assemble them and then we can use phylogenetics to compare them to each
Yeah,
other.
to understand the diversity of that space.
Or you can just name it taxa 1, taxa 2, like who cares about the name in the database? But,
Right, and then you have an idea of how much diversity is there as opposed to just unmapped reads.
right, yeah. Yeah, it's interesting, depending on what kind of collaboration you're in, what people want is just like, they want a biomarker. And it's interesting to make an analysis that is interpretable to the people around you. But no, I totally see your point, and it's a good one. Yeah. Awesome. Are there any other?
Actually, yeah, because, I know I already asked the question, but you had another slide that had segments that are associated with the breakdown of fiber, and something stuck out from that graph, which is that the Tanzanian population, I work in Tanzania with Hadza hunter-gatherers.
Oh, you do?
Yeah, and that really struck me because when we do the dietary analysis in terms of what foods people are bringing into camp, looking at what people are eating, just looking at the gross content of the foods they acquire, typically you see a huge amount of fiber. So
Yeah.
our understanding from that perspective is that they're eating a ton of dietary fiber.
Yeah.
So I was really struck by this result.
Yeah, and so this is the opposite of my expectation, although I do know there's seasonal variation and that there's some seasons with less fiber. In my mind, this might reflect the sequencing depth, but I also happen to know that like the Sonnenbergs have been sequencing those samples really, really deeply. But I don't think we have their most deep data set. I think this might be shallower sequencing. So some of this could reflect the sequencing depth. but overall, I just have to agree with you. That surprises me too. I mean, my expectation would have been that it would be higher. I know there's other populations where these are cazymes, the carbohydrate breakdown enzymes. The diversity is lower than expected. Like I've heard of an Ethiopian cohort that has just really, really low cazyme diversity, and apparently it's a very milk-rich diet. And so maybe, I don't know, there could be things that change the richness of those genes. Or they could be unknowns. I mean, yeah, but that surprises me too. I totally agree.
Same graph. Do you know why the other U.S. sample is so high?
That could be sequencing depth related. This is I should go back and review exactly which cohort this is. I think this is a cohort of men with HIV, but I should double check it. And I think it was pretty well sequenced. And you know, I don't know why that one is higher. I mean, this is science, and I have to admit, a lot of the results that came out as we did this analysis were surprising to me. So I guess that's the main thing I can say. But yeah, it also might have to do with the sequencing depth and the particular genes that we included in the analysis. So, but yeah, that one definitely sticks out as surprising.
I'm wondering if there are any samples from communities that have like more recently shifts in food changes, I guess.
Mm-hmm.
So like I'm thinking of, I read a book, The Weight of Obesity in Guatemala, and it was about traditional foodways that have like recently been changed and industrialized. And I'm wondering, like they talked a lot about the industrialized version of what nutrition is compared to like traditional, like they just maybe don't use measuring cups or don't think of nutrition the same way. And so I'm wondering if there are any samples from like recent shifts in those diets.
Yeah, that's a really cool question, and I've brought up the Sonnenbergs up at Stanford a few times, and that comes to mind immediately because they have a very interesting cohort that has a gradient of industrialization in Nepal, where it's villages that have different access to processed foods essentially, and you see a gradient of microbial diversity. In their data set, it's not surprising. It's like exactly as you would expect was my interpretation of their paper, which is published, and I'm happy to send it around. So that I have seen examples like that comes to mind. And in general, as people get more access to processed food and are eating less fiber, you see these carbohydrate enzymes become less abundant in the gut microbiome, and the amount of Prevotella goes down. Or actually, that story I told about the Thai immigrants is a really good example, because Prevotellas are really rich in these enzymes. So with every 5 years people lived in the US, those microbes decreased. Yeah, cool question though.
Thank you for the talk, Professor. I'm wondering if you have observed any form of microbial metabolite profiles evolving over time, you know, given like the regional differences and then the elevations, but also at the different fermentation time points. I'm wondering if you have looked for that kind of stuff, like, you know, during, midpoint, and after the thing has been fermented, and if you have seen any forms of signaling in the metabolite profiles that may potentially talk about that. For example, in
Do you mean in the foods being fermented?
Yes, in the fruit and the chicha, yeah.
Oh yeah, we haven't done like metabolomics or any other analysis with those samples, but that's a really good question, and it certainly should be possible. And I know there's big efforts to use metabolomic analysis for all different kinds of foods. Down in San Diego, for example, Peter Dorstein's group has a global food project where they're using metabolomics to characterize every kind of food they can get their hands on. And fermented foods are such an interesting one to examine. Clearly, those molecules have really big health impact and microbiome impact. And when people have compared diets that have fermented foods plus fiber versus just fiber on its own as an intervention, the fermented foods help the microbiome in the gut diversity go up, which maybe shouldn't be considered a measure of health. I appreciate that comment, but in general, people eating fermented foods and fiber, their microbiome diversity goes up more universally, whereas fiber on its own tends to have a more individual response. And another thing that's really interesting about that to me is that it's not that the microbes in the fermented foods are colonizing the gut, they tend to be transient. It's thought that it's the molecules in the food that are cultivating maybe lower abundance taxa that were below the limit of detection, or maybe new taxa that come in and then are able to thrive because those molecules are there to cultivate them. So those molecules are fascinating. That's a good question.
I'm curious about the cleanliness in the U.S. today. A lot cleaner than it was when I grew up.
Yeah.
Does that have an effect on all these microbes?
I mean, so the question is about cleanliness, and I would assume it does. I mean, it has to. That's a really good question. My own thinking about that honestly has changed in the last few years. I used to feel that what we need are human microbes or fermented foods that help cultivate human microbes, and so the microbes from the soil or other environments would just be transient anyway, so what influence could they really have? And one study that really affected my opinion was from a Finnish group. There's an institute that has done a series of studies, one where they took a kindergarten that, if you looked at a picture of it in 1920, it was like a farm with flowers and soil. And then if you looked at a picture of it in 2020, it was like concrete. So they took some of the concrete out and made it look more like it did a century ago. And then they took microbiome samples from a bunch of kids in the kindergarten before and after, just 1 month after that intervention. And they saw really interesting changes in their microbial communities and in their immune markers. And I'm happy to find this paper if it's interesting to you. There was another really similar one that used green walls. And my lab got really interested in this. We're actually gonna try to make a green wall in our lab as we're having a spring greening instead of a spring cleaning. And this green wall, it's out of our budget, but this study used a Dutch company's green walls, and they have a hydroponic system with plants growing, and then air filtration that funnels the air across the plants. And so it's like a HVAC except with plants. And then they put these into office buildings and hospitals. And they showed, especially in the office buildings with with not as good circulation in comparison to hospitals that were like really off the charts with their HVAC, that the people who worked in the office building with the green wall also saw increases in microbial diversity, which is a tricky measure. I keep coming back to it and pretending that that's an obvious signal of health, but that was what they saw. They saw more microbial diversity, and I think they had some immune measures in that study too, so I can find that one too. So those are two pieces of information that influenced my thinking about that even if the soil microbes are not adapted to the human gut and don't immediately colonize you, they might still help promote the growth of other microbes that could. So, yeah.
Wonderful. We've reached the end of our time. Thank you, Katrina.
Yeah, thank you.